my first engagement with psychiatry as a discipline. I had done my dissertation at Minnesota on alcoholism among American Indian folks living on the streets in Minneapolis, but I had not really interacted much with psychiatry. And I was here for 13 years before I got the job in Oklahoma. And almost immediately, in fact, the very first faculty meeting I went to in psychiatry, Bob MD came up to me and introduced himself and said that he understood that I was interested in narrative. And I had an interest in narrative. It turned out Bob was interested in the narratives of 4-year-olds. I was interested in the narratives of alcoholics and former alcoholics as they tried to make sense of their experience. But we clicked immediately, and it's been a collaboration that's lasted through my time in Oklahoma. Bob is a very valuable collaborator, and he's responsible for the work that I want to talk to you about today and tomorrow that we've been doing around the genome in Native communities. Bob had developed an interest in genetics, was working with geneticists to understand development and understand individual differences in development. And so I started to think, well, that might be interesting for us to pursue too. And I knew the knowledge was controversial in Native communities. So we entered with some trepidation. But I got involved in a field that I'll talk to you in a little bit about, which is called ELSI, E-L-S-I, which stands for Ethical, Legal, Social Implications Research. And I was so new to the field when we submitted our first grant that as I was calling around trying to identify consultants to come help us, people would complain about ELSI, and I didn't know what they were talking about because I assumed that I didn't know you could pronounce it. So that's how new we were to the field when we entered into it. But I got to know a guy named Morris Foster who recruited me to OU. And then subsequently left to become the VPR at Old Dominion. But almost all of my work now at the University of Oklahoma is focused on genomics and the implications of genomics for Native people, both in terms of clinical and translational research generally. There's not a field of medicine that doesn't have fantasies about what genetics is going to mean for revolutionizing healthcare and cancer specifically, where I think arguably we have seen real clinical utility from genomic knowledge, and in tribal health systems where we have partners who are really interested in working with us to articulate knowledge in ways that respect tribal traditions and that can generate clinical work that's of value. So what I want to do today is describe a movement in biological sciences away from raw sequence data, which has been the preoccupation of the Human Genome Institute, remains a preoccupation of the Human Genome Research Institute, toward a much broader range of approaches to the molecules of life that allow us to really reconnect in some much more rich ways with the kinds of concerns that I have as a social and behavioral scientist, that raw sequence data simply doesn't. It's quite disappointing in multiple respects. I'm not a geneticist, I'm not a biologist, I have no formal training in microbiology or chemistry, so I'm going to travel lightly over my collaborations in those areas. But what I want to show is the opportunities for real rich interdisciplinary work and dialogue in the opportunities that are created there. The central problem is, I think, quite familiar to the DPRG audience, right? There is a gap, a significant gap between the preoccupations of molecular bioscientists and the interests of social and behavioral researchers like myself. There have been tremendous advances at, more interstitial areas. I think of the neurosciences, for example, where you have a proliferation. All sorts of different kinds of neuroscience now exist that reflect their engagement with social and behavioral processes, affective, cognitive, social, interpersonal. All of these kinds of neurosciences reflect our ability to link the brain to its social and behavioral context. But that integration has proven much harder at a molecular level. This is a persistent tension at every cancer center in the country, where the scientists are the bench scientists working on molecules, and those of us who are out in cancer prevention and control are doing something that's not even understood as science, let alone something that can enter into dialogue with that molecular research. But the new approaches that are enabled by the revolution we've seen in the life sciences and by bioinformatics, I think, open up really exciting possibilities for collaboration. But as I'll say when I close, I don't think we need to stop there. I think we can take what I think is an ecological perspective and move it up a level and out into communities and out into neighborhoods. I'm gonna, since we started late and we maybe I want to make sure to leave a little time for questions, I'm just gonna run through this briefly. We have been involved in ELSI work at almost every phase of this sequence of core genomic resource generating activities from the Genome Institute. The Human Genome Project was actually a human genome project. Although there was more than one individual, it was largely just an attempt to describe what was understood to be the base sequence. The Human Genome Diversity Project got off to a notorious bad start with Native people when they indicated their interest in rescuing DNA before the people went extinct. You can imagine the reception they got in Native communities with that kind of entree. But that generated an interest and reflected an interest in human genomic diversity, differences between people, differences between genomes. Our first ELSI grant was funded under the effort to develop the Polymorphism Discovery Resource, which is a federal pharmaceutical partnership to generate a database of single nucleotide polymorphisms. We continued to do that work under the effort to create the haplotype map, which was to understand the architecture of the underlying sequence, the things that are correlated with each other. All of this work has been focused on population structure and data resources on the underlying human DNA. Nothing about when it comes to life, how it gets transcribed, how it comes to make human beings. None of that, just the alphabet of the human sequence. Each of these genome research projects has had an ELSI component, an ethical, legal, social implications component. It's been a part of the sequencing effort from the beginning. In fact, many of us see it as a PR stunt by Watson to clear space, understanding the knowledge was going to be controversial. He set aside a portion of the sequencing dollars to address public concerns, which we would generally assume he saw as silly and uninformed. And ELSI has been a part of the sequencing effort from the beginning. Genome scientists, as I said, when I was calling them in the context of my first ELSI project to get them to the table, they were complaining. They wanted that 5% as well. They didn't understand the value of devoting that to this thing that they were pronouncing as ELSI. But it's now the biggest funder of bioethics research in the country. And genomic concerns and genomic questions have come to dominate funded bioethical research, at least in our country. Almost all of it is focused on what genetic knowledge reveals most clearly, the ways in which we're connected to each other. And so a lot of it has focused on the implications of genetic test results for other family members who may or may not have understood that they were participating in a genetic protocol or getting a genetic test, but by virtue of their relationship to the person who did, are implicated in that and receive that, the knowledge is generated there, but also for populations. There's been a lot of concern about the implications of genetic knowledge for identified populations. And it's a focus on heritable differences. That's almost exclusively what we get in all of those efforts that I described previously. It's almost all about population structure and heritable differences, both between populations, so the ways in which populations differ from each other, surprise, they don't differ as much from each other as any individual within a population does to any other individual, but also between those individuals within populations. And almost all of this work has really been haunted by two really significant abuses of genetic knowledge in the past, or knowledge of heredity, or purported knowledge of heredity in the past. The idea that we could improve the species by manipulating breeding, eugenics, and the idea that they're genetically inferior populations, which I think most of us would understand as racist. Just a few words about Elizabeth Warren. She's in the news. The Cherokee Nation does not have a biological criteria for membership. This test is never going to prove, and the tribe made clear yesterday, this test is never going to prove she's a member of the Cherokee Nation. Obviously, it was a response to Trump's taunting her to do it, claiming that she had no Native ancestry, like a lot of Oklahomans who look very white. There's undoubtedly Native ancestry in a lot of people's families. That's what she said, and that's what she's proven. But she's doing it in a way that plays to Trump's attempt to racialize tribal identity. And it's potentially quite dangerous to tribal sovereignty. Tribes insist they have the right to define members any way they want, and it's generally not biological, certainly in Eastern Oklahoma. But clearly her family lore is not a complete fabrication. And indeed, those of you who've seen the ads on TV know that one of the early applications of sequence technology is in ancestry testing, the kind of recreational stuff that maybe some of you in the room have participated in. My wife has dragged me into some of it as well. And it's probably one of the biggest applications we see today of genetic technology, and it's all commercial. Clinical utility of sequence data has been disappointing. That hope for revolution that Francis Collins still won't shut up about the impact of genomics on healthcare, has not materialized, and it has generated significant backlash and concern about the ways in which this knowledge has been hidden. There have been important insights into rare heritable disorders, of course. It would be idiotic to dismiss that. And I think we've seen revolutions in cancer care based on understandings of what is going on in the tumor that are genetic in origin. But there's been little clinical progress. It's based on genetic knowledge on most common chronic diseases, what we should eat right, we should eat better, we should exercise, these sorts of things. I didn't need the genome to tell me, right? And the genome doesn't help me do it either. And the integration of genomic information into healthcare has been slow. I mean, we struggle already with electronic medical records, and the idea that we're going to add a bunch of genomic information in there and clinicians are going to be able to use it in the 15 minutes they have with patients is, I think we should be suspicious of that. But the hope or the hype for genetics springs eternal. And as I say, Francis Collins is a source of that. And it's indexed in a major new initiative to develop a longitudinal cohort of 1 million people. It was called the Precision Medicine Initiative for years. It's now called All of Us. The goal is to combine sequence data with electronic health records, sensor data, ecological momentary assessment. It envisions a crowd probably much like a group of participants, probably much like many of us in the audience who have wearables, who are interested in our health record and interested in providing sequence data. We've already done it to commercial enterprises for ancestry testing, that are engaged in a dialogue with participants. When I, as that kind of person, listen to the promotion of All of Us, I get excited. I would participate. I have to tell you, most of my colleagues in ELSI would not, for reasons that I find concerning, right? The fact that the ethicists don't want to participate in it, including the people who are charged with protecting the health record information. But it's exciting to me. It's the kind of thing that someone who's interested in that kind of approach would be interested in. That's people much like myself, but that is hardly representative of the US population, let alone the global population. And so there are persistent concerns that I'll talk more about tomorrow about the extent to which All of Us was going to realize any of its goals with regard to diversity and inclusion. And that's a persistent problem in the genomic resources of this country. It's almost all Euro-American, and when the Asians got involved, Asian countries got involved, there was a growth of Asian samples. But there's virtually no Native American samples from either North or South America, virtually no African samples in any of these resources. But maybe this will yield the benefit, the hoped for benefit of all this investment we've made in bioinformatics. But as I said, we may already be there. We're just looking in the wrong place. There has been a revolution in the life sciences that is enabled by the catalogs of molecules that we've been able to accumulate. We're literally awash in omics. There's a proliferation of the term. We add it to everything now, right? And basically, it just means the totality of something, right? So the genome is the totality of your genes. We have transcriptomics. We're started to look at gene expression. We have proteomics. We're starting to look at the products of gene expression. We have metabolomics. We're looking at all the small molecules that are at work breaking things down in our bodies. And it's now generally glossed as systems biology, by which we mean the bioinformatic approach to the contemporary life sciences, almost exclusively preoccupied with molecules. I want to talk to you a little bit about our work on the Human Microbiome Project. When I moved to Oklahoma, it was just getting started. And of course, it had an ELSI component, like all of these big initiatives do. And I remember seeing the funding opportunities, and I said, there's something even I won't do. Because I got accustomed when the time I was here, it was completely soft money funded post, to writing a lot of grants on things that I didn't really necessarily care about or weren't necessarily integral to what I was trying to accomplish. But almost as soon as I landed at OU, I learned that my colleague, Cecil Lewis, who's a geneticist, was really interested in the microbiome and in microbiome diversity in particular, sampling from rare and unincluded populations, unlike St. Louis and Houston, where the core HMP work was done. And so I guess you should never say never, right? Because I found myself doing that, and that's led to the work that I'm here to talk to you about here. And I did that work both with Cecil and with Morris. Yeah, need to fix something. All right, I'm all right. Our entree into the work, the reason that communities were interested in this work is that they've long been aware that dietary change has resulted in chronic disease in populations. And so we were able to enter into a dialogue with communities, not about, hey, you want to participate in the generation of resources for bioinformatic work, but instead on a way of connecting to, I can stop, you can fix it. Yeah. Having trouble with the projection. All right, people can't see my screen. I'm just repeating myself. Yeah, 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 whatever you need to do. At any rate, our entree into this was for on diet and disease. Yeah. That sets the mood. So we had to cast about in Oklahoma for a time to find a partner for this work. People are like, hey, you want to help us with genetic research? People were like, no, no, thank you, we don't. But ultimately, we were able to partner with the Cheyenne Arapaho tribe in Oklahoma. It's a group of merged tribes from the 19th century as a product of how tribal populations generally ended up in Oklahoma. And we spent 3 years engaging the participants before we sampled at all. So these were the most thoroughly informed participants you could imagine participating in a study that involved fecal fecal samples and saliva samples. We were not focused on human DNA. We get to those issues later. But we spent 3 years working at the tribal level in terms of tribal health leadership, making sure that the tribal leaders understood what was going on. We worked across the jurisdiction, so we worked in different communities with different leaders in each of those communities, and we worked at the individual level to make sure that the participants understood what we were doing. And then, as happens with this stuff, you've got the samples, you've got a new technique, you might as well just do it instead of following that method, which is standard in the life sciences. We went back to the participants and talked to them about our interest in doing additional analyses of the metabolites. And got their consent before doing that. And that made my collaborators in the life sciences really nervous. They're like, we got it, man. We can do what we want with it now. And we're like, no, that's not the relationship we have with the participants. It's not the relationship we have with the tribes. And that represented a compromise and departure from how they would normally do things, but one that ultimately worked just fine. The findings broadly, we saw there was a substantial overlap. There was a Norman sample that was non-native. There was a sample. I will stop for that. My whole family's in the Norman sample. And then there was a comparison with several Peruvian samples. There were differences in the family level between Norman and Cheyenne Arapaho communities, family level of the microbiota. There were additional differences between the metabolites. Most of the diversity, though, was not between Norman and Cheyenne Arapaho, but between Oklahoma and Peru, where there was much more diversity in the Peruvian samples that were either traditional agriculturalists or hunting and gathering people. And the metabolites are the easiest to associate with disease and give us the most insight into what may be going on as a product of the ecologies in which people live. And we'll be, for those of you who will come to the seminar this afternoon, we'll be talking about the paper that we published. What was interesting about this is a biological journal, Current Biology. They were as interested in our community engagement and in the social and behavioral data that we had as they were in the biological data. This suggested to us that we could use existing bioinformatic resources to link to and understand the social and behavioral and environmental determinants of health disparities, that is diet and lifestyle, giving us a very real example of how inequality shaped our biology at the gut level, right? We often say the ways in which inequality gets under our skin, right? In this case, it's largely on our skin, the microbiota, but still, the ways in which it affects our biology. And it was suggestive of a bunch of additional studies that we're pursuing now with the tribe, both in biological and social and behavioral work. Cause and effect are really far from clear here, right? If we had mice and we could transplant gut microbiota, maybe we would see the causal relationships you see in mice. But there's an equally compelling suggestion that what you see in the gut microbiota is an exhaust indicator. It's an indication of what's been put in to the body rather than a pathological biological process. Or is it both? And in all likelihood, it's probably both. But regardless, there's a whole bunch of interesting questions that we're able to pursue at both a molecular and a social level. So our work on the gut microbiome raises questions about food, what's available, what people choose to eat. You have to really work hard in Oklahoma. I know, having lived there now for 10 years in a much more cosmopolitan environment than participants from Cheyenne Arapaho tribes, that it takes a lot of work to eat healthy in Oklahoma. That's true in America in general, but it's certainly true in environments like this. And equally significant, what are the biological systems that this gut is interacting with and shaping? and the opportunities to connect those two streams in future work. But not surprisingly, this is technology that's broadly available to folks. People, you can sequence anything. There's lots of sequencing capacity in the country. People are not interested in that kind of exploratory work. Increasingly, study sections are demanding, especially about microbiome work, but as true in, I think, genome work. The specification of a mechanism, the testing of a mechanism. And so we really need to grapple with that in order to get funded for the next step. We need specific hypotheses and mechanisms. I think we're all interested in epigenetics. It's been in the media. There's a lot of attention to it. Epigenetics in the new sense of the impact of information on the DNA and its expression, especially the idea that these changes are heritable. This has gotten all sorts of attention in Indian country. The idea that trauma is transmitted biologically across the generations has really caught fire. But we don't need to go that far to understand the importance of gene expression. All of us are here not because of the core sequence, but what's been built of that core sequence. And our interactions with environments shape the chemistry of our bodies in some pretty fundamental ways that can be measured at a molecular level, including products of our own and all the other cells that are with us for the ride. They certainly affect children in utero, so you can get what appears to be a heritable difference. And but they can also be transmitted environmentally through transmission of microbes, which we share with each other. I'm sharing mine with you right now. I brought some from Oklahoma. I hope you enjoy them. And those kinds of transmissions are important to understand as well. And I think what this points to is the importance of a broader ecological perspective. And as I'll say in a sec, that's not new to people in this room or to developmental science in general. But I think there's some new approaches that are opened up. When we arrived at OU, we didn't know they were creating a doctoral program that was designed to blend cultural and biological anthropology. There's a lot of things they don't tell you when they're recruiting you, right? that you might want to ask about for those of you who will soon be looking for work. And what they created was, what we ended up creating was an integrated biological and cultural program that we now call Human Health and Biology. It's both my colleagues in genetics and us on the medical side, but it's plagued by the same tensions that I began this talk with, that there are few opportunities for dialogue between that molecular level and the social and behavioral preoccupations that most of us on the other side have. But one of the very real opportunities that exists for this is an ecological work, which is a core commitment of any anthropologist. There's not an anthropologist out there who doesn't believe in fundamentally ecological approaches to problems. And as I said, this is not news to anybody in developmental science. Here at Bronfenbrenner, now 50-some years ago, articulated a perspective with all sorts of levels that I don't bother to remember anymore about the systems and their impact on individuals and the way in which we are nested within multiple systems. But new molecular techniques and the resources really allow for many deeper analyses. And perhaps most importantly, they open up biology to that kind of inquiry. Now, ecology has been a part of biology for years, forever too. All of my colleagues in biology in Oklahoma work on stream ecology. They don't work on humans at all. So I'm not saying that ecology would be new to biology either, but the opportunities that bioinformatics gives us to interact and understand interactions with context, I think, are significant. This is where I go completely off the rails and talk about something completely different. This is a project I would never have engaged in had I been here, because it was funded by an R03, which would provide minimal support to either the investigators or anybody else I was trying to fund. But soon after we landed in Oklahoma, there was an opportunity from NICHD to look at research on human-animal interaction. It was co-sponsored by Waltham, I think clearly designed to stimulate pet ownership and pet feeding in particular, and animal-assisted therapy in particular was the focus. They really wanted to generate clinical trials on animal-assisted therapy to understand the impact. But we snuck in a proposal on our second try to look at roaming dogs up in Montana, the Northern Plains Reservation. And we focused on their impact on children. This is indeed the global norm. But what was motivating us for the roaming dogs are the global norm. What was motivating us in this work was concern in the community that my wife had come to know. That dog suffering and loss was really making children callous or indifferent to suffering and loss more generally. They were stepping over or dead dogs or injured dogs, ignoring injured dogs. But it turned out that the community really didn't devalue canine life in the ways that people thought. That they certainly were aware of the suffering, they were affected by the suffering. They had a really unique relationship with dogs. They were revered, unlike all of the other tribes of the Northern Plains. They were not consuming dog in ceremonial context. But the dogs were generally let to roam with predictable consequences in environments with cars and other dogs and people. Children and the homeless, we did a series of year-long systematic community observations. You never saw groups of children or homeless without dogs. They were there with them, constant companions in this context, but they were seldom the subject of direct interaction. I was telling Bob, we've rebuilt our stock of pets in my home now, and we talk to those animals every day, and we baby them, we interact with them. That's not what you see on the streets of this reservation, right? The dogs are there, they're valued. It turns out they're loved and missed when they're gone, but people are not interacting with them in those ways. In fact, what we discovered in our interviews was great love for dogs. They were lovingly imported to the reservation. Puppies don't stand a chance in environments like this, right? So you bring in adult dogs if you want them. Certain community dogs were legendary. Everybody had memories of certain dogs in the community that had an impact on them. And there was palpable grief in families when they lost their dogs. You know, from my perspective as a suburban homeowner who has a fenced yard and walks my dogs on a leash, I was like, well, maybe you should do that. You could protect your dogs, right? But there was that kind of emotional connection to dogs that you do not restrain is the global norm. This is probably how we co-evolved as species. That's both for good in terms of feeding and care, companionship and protection, and also for bad in that those dogs get attacked or those dogs do attack. And also get lost. You lose them to attacks or injuries or cars. But the ways in which this is manifest, I think there's important lessons here for how we approach parent-child interaction cross-culturally. The ways in which this is manifest are not what we would expect to see based on our ideas of suburban pet ownership. And the reason I digress there is just because I think it's interesting. People like our work on dogs, and it is ecological, certainly, is that I think it points to a broader perspective on what we might think is possible with regard to systems biology, which is almost always seen as molecular. The view from Indian country, as I said, suggests a broader perspective on systems and interactions, including the quality of food, the impact of colonialism. But this is one that I think would allow us to much more effectively bridge a charge that we were given in the 90s in a report that Bob was an author of bridging work between neurons and neighborhoods. I was looking for an M to complement molecular, right? Because we're not even talking about cells in a lot of this work. We're talking about molecules, but I didn't have the M at the other end. It's a mesoscale. I should have said molecules to mesoscale, right? So at any rate, systems biology creates, I think, new opportunities for us to advance biological work on health disparities that has not, as I said, traditionally been the focus of this molecular work. The core samples of the HMP were recruited in St. Louis and Houston with populations that had no chronic disease, very unrepresentative even of Houston and St. Louis, let alone the rest of the world or the rest of the country. Focus on gene expression and its biological context really opens us up to work on the environment in a way that the human genome never would. And it might serve to inspire even broader ecological knowledge of the sort that we've been doing with dogs. What I want to talk about tomorrow are the opportunities that this specifically has given us for engaging Native communities. The opposition of Native communities to the articulation of genomic knowledge is real, persistent, and an obstacle to that kind of diversity and inclusion that I talked about in genomic resources. And this kind of approach that allows us to link biological work to the concerns of communities really gives us an opportunity for community engagement that NHGRI has yet to realize and certainly has yet to actually implement in any of its initiatives. So let me just quickly close with some acknowledgments, and I have left some time for questions. I want to thank Bob, MD, for the invitation and for the funding, the Department of Psychiatry and your fellowship, for the hospitality. And then we've had, and I'm sorry, I forgot I added the NICHD grant at the end, but both the NHGRI and NICHD for funding, including our work on the HMP, which was an R01, our work with our CEER, our Center for Excellence in ELSI Research, which was a roadmap initiative, and the R03, which was the work on dogs. So thank you for your time. And I can take questions. I don't know if we ever got the slides showing, but I was largely reading them, so hopefully they got it. Yeah. 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 Mark. Mm-hmm. We're probably bothered by the dogs. Yeah. 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 Well, this is up in Montana where we're doing this work. Well, and both are true, right? I mean, these dogs are a nuisance for sure. On Navajo Nation, where I did work for years, dogs are very much a nuisance, are not generally celebrated or enjoyed. What's unique about this community is that there really is a lot of ambivalence, right? There's close connection and respect for the dog. People are bringing those dogs in. Some of those dogs are quote unquote owned. They're seen as connected to a family, but they generate problems too. This is a community that's not immune to fatal dog attacks. They happen. They're rare. They're remembered in legendary form. But yeah, I mean, roundups are not uncommon throughout the reservations of America, where dogs are just brought together and killed. So it's both, I think. But what's unique is what's the norm is roaming dogs. So it's not normal to keep your dog behind a fence and with a collar and a leash, right? But it is normal for you to have a dog that you have a connection to, that you have a relationship with, and that you would mourn if it was lost or if it was attacked by other dogs. But that doesn't mean that they don't become a nuisance or worse either. So right. So there's a lot of people feeding dogs. There's beds at people's homes where dogs can come back to sleep. But who knows when they're coming home, right? So there's much less control over the dogs for sure, which generates the problems that you described. So I don't know if it is that different. There's a recent book on dogs at a dump in Mexico called "What Is a Dog." Where the folks that are working at that dump are feeding these dogs, caring for these dogs, but these dogs are getting in the way. They're snapping at them. There's all sorts of problems that come with that kind of approach to canine life as well. But there's no doubt that that is globally the norm. That what we do in the suburbs is not normal. Right? It's yet another example of weird interactions that Western educated industrialized rich democratic societies, right, which we tend to assume is human nature is anything but, probably in fuller context. Is that Larry? Is that Larry? Yeah, I thought I recognized you. How you doing? Yeah, 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 yeah. Right, right. Yeah, well, so I mean, and I was talking to Mark about this before we got started. I think if you approached the issues often are not at the individual participant level. people are interested in what their genomes and what that information might reveal. The issues are more at a political level, where people are trying to control what is known, how the information is used. And I think really the way forward is to work at that level to create formal agreements and infrastructure for that research, as you are doing, I think, or could do in the context of the work you described. But people were not at the time identified with their microbes. There's a classic paper that Kieran O'Doherty did, Who Owns Your Poop? Right? Because it points to some of the ambiguity. It's not normally something we're trying to keep around or hold on to, right? Unlike maybe our sequence data, which might feel a little more personal, a little more protected. And so people weren't concerned about it. And really the energy was in understanding what was going wrong with their health as a result of dietary change. And so it was the opportunity that gave us, we also paired it with some analyses, attempted analyses of ancient gut microbes from coprolites. That didn't really work because they're mostly degraded into soil, and what you see is not a profile that's consistent with gut in a lot of those samples. But the idea was we could maybe look historically, we could understand more of that. People were really engaged in that process. Leadership wasn't concerned about it. And I think, again, I think it's because it wasn't identified as theirs. But I think there are ways forward with the human sequence data as well. But it really relies on more equitable partnerships, more transparency, and restrictions on data sharing, quite honestly, which I know runs counter to a lot of genomic approaches, right? But it's probably essential to doing that. We have a pending proposal as part of a collaboration of cancer centers down in the southern and southwestern U.S., where we've created data enclave to start to look at tumor genetics, especially in kidney cancer, where we have really dramatic disparities and nobody has any idea why, as opposed to colorectal, where you could point to screening and diet, or lung, where you could point to the rates of smoking. The kidney cancer is mysterious and probably does reflect something of underlying genetic vulnerability. And we're proposing to create an enclave. That's with a corporate partner, not a federal partner. So we'll see. But NCI has indicated flexibility on this issue. The one that's harder is NHLBI, and even NHGRI has indicated flexibility because they want this work to proceed. They want to train the next generation of researchers, and they recognize that they can't just do it the way they've done it. So, but yeah, people were not so concerned about keeping their poop or protecting what was done with it. Yeah, so we have and that was the same. People were as willing to part with those. There was also some skin sampling that I've never seen the results of. But it was, but we've published both the oral and the fecal work, and there was not a problem with that. But again, that's because people, number one, they were interested, and we had provided assurances that we weren't going to look at who they were. We were going to look at who was along with. Yeah. Bob, sorry.
One of the fundamental issues concerning development has to do with understanding the mechanisms of change over time during individuals or groups' lifetime. And it's striking to me, I will remark, that in your presentation and discussion, you didn't mention much about cultural dynamics
Mm-hmm.
in relation to aims and thoughts. So a specific question would be, when you describe
All of us. Yeah,
the
this million-person cohort. Yeah. Yeah.
question would be, what are your hopes and expectations and your personal aims of your work to study cultural dynamics in relation to the epigenetic changes across the lifespan?
Yeah. Yeah. So I'm not involved in all of us, and honestly, I'm glad I'm not, because I think they've made some serious missteps in tribal communities. But Spiro headed their tribal work group and created a position paper that I think is quite respectable. It's too late for it to impact the way they're proceeding with data collection. Yeah, it's published on the web for all of us, and all of us is recruiting. They're generally recruiting through health systems, but they are not. Yeah, Spiro Manson. Sorry, yeah, that was a little too intimate there, right? But at any rate, all of us is not recruiting for community and cultural dynamics, and the sampling strategies don't involve that. There may be some environmental sampling at some point. Undoubtedly there are if someone will pay for it. But it's largely, as I said, focused on linking the medical record to the sequence data and doing ecological momentary assessment and using sensors to understand activity. So there's potential for looking at neighborhood, community, geographic kinds of dynamics. But the cultural changes of the sort that have shaped canine interactions with canines in cluster housing and the sort that's changed the diet of Cheyenne Arapaho folks, that there's not that kind of time depth, and I think it's largely individual level work that's contemplated. But I'm not involved in it. But I think you're right that what was so exciting for us was I took the HMP, which, as I told you, when I moved to Oklahoma, I couldn't imagine anything less relevant to where I wanted to go with my career, and it turned out to be quite central and quite interesting to community partners because of the opportunity to understand dietary change. Yeah, we do. He came all the way from Israel for this talk. Yeah, thank you for coming. Yeah, you didn't actually come all the way from not directly, right? But I appreciate you coming. Yeah. So, as I said, it feels a bit like preaching to the choir here. Obviously, you guys are doing sophisticated biological work in the context of social and behavioral rigor. But I think that there's all sorts of opportunities that really from a life science perspective to connect with life as it's lived experientially and socially that are really new and very interesting. And there's undoubtedly all sorts of opportunities. No other questions?
We had a presentation last year on Owen's platform.
Yeah. Again, yeah, I'm going to defer to the experts in the room. Yeah.
One of the challenges levels
Mm-hmm.
of function in terms of regulatory systems.
Yeah.
At that level, I found it DPRG, especially the excerpt themselves,
Yeah.
as far as that.
Yeah.
But it's a similar problem throughout all
Yeah.
as usual trading.
Yeah.
You're working in all the way up to
Right. Yeah, I think that, I mean, those resources are there. I'm sure they're being refined daily, if not hourly. But it's no longer sufficient to just contribute to those resources. The NIH and our quote unquote peers aren't interested in funding that kind of exploratory work. They want us to do something to build hypotheses and understand mechanisms now that those resources are available. And a sure way to do that is to be able to connect to the kinds of concerns that we have on social and behavioral processes that would allow us to do that. And I mean, I wasn't kidding about it would be nice to get below the cellular level, the neurons that characterized the report, the base level of the report that you were an author on, Bob, to get down to that molecular level. That's itself a gap, right? Let alone the kind of gap that we're talking about on the other side. Oh, nice. You know, like molecules to mesosystems. We'll work on this over the break. Yeah.
stress research, cellular or mobile information levels, as well as behavior, social systems, and clinical. But I wonder how
Well, one of the things I was thinking about is I discovered this concept 3 or 4 years ago. It was something called complexity science, an approach to hospitals and medical care, which is basically what you're talking about. It's not a simple implementation. You should know this. There are a lot system would be modulated by psychological factors, regardless of what the ethologists thought 30 years ago when I started postdoc. It was no surprise to me. And we've actually had positive changes in inflammatory pathways, transcription pathways, found that psychological interventions can actually appear to affect and modulate those transcription pathways in a way that's positive for including organisms, something that's negative or stressful. So
Right.
it's not simple. And I remember when the first data set was sent to me by UCLA, sent me this spreadsheet that had 27,000 variables per participant times 2, which were the pathways he was looking at. It moves to a level of analysis, which is something called self-speed UCLA, to actually disentangle this and identify inflammatory pathways, oxidative pathways, any of these sorts of things which are relevant to those of us in the stress field. So for him, it was relatively simple. For me, it was mind-boggling. And it's the same thing with looking at microbiome. It's just, the numbers of organisms vary. It's what, 100,000 or more, or something like that.
Yeah, I don't. I mean, at the species level, I have no idea.
Yeah, and you see things like a new person taking a whole microbiome shift.
Yeah.
Or you get a new pet and the whole microbiome shifts. Or you take an antibiotic, the whole microbiome shifts, but it's regionally specific, where someone had an infection, for instance,
Yeah.
somebody has an ear infection, and see these antibiotics targeting the culprits that are creating some of the infection instead of replacing it.
Yeah. There was a question back here. Or comment. Yeah, I mean, I'm sure everybody in the room has an opinion on this, but I like, I mean, I skipped high school chemistry. I never took it in college. I'm going back to try and understand chemistry now because I like to be able to understand what my colleagues are talking about. Ditto with microbiology, but I'm not going to become a chemist or a microbiologist. At least I don't think so. I think that much time left. And so what I think we need are people who are able to engage in that kind of dialogue and articulate protocols that are mutually beneficial. And I don't think either of us are really necessarily in my generation well prepared to do that, right? Molecular folks are going to do their molecular work, and they don't even see what I do as science, right? I skipped high school science classes, right? I don't understand what they're doing either. And so I think what we need is some basic level of literacy that would allow us to communicate between the two. Maybe there's some specialists we can create in between. I think certainly DPRG creates a certain kind of approach that is interdisciplinary within the person. But I think it's more and more, it has to be done teams. And I think, as I said, my colleagues who do the bioinformatic work, I mean, they need hypotheses, right? They're not getting funded anymore to just do exploratory work. And so they really need to have ideas that come from that kind of conversation.
So I think one of the things one needs to teach is collaboration.
Yeah.
And that's, we work in towers within the university and become really isolated. That's not going to work.
And I will say, you guys have it so much better here than we do in a classic college environment where departments are real entities and fighting with each other, and they're training people in that discipline. Damn it! And yeah. So whereas here, I think NIH largely forces interdisciplinary collaboration in some ways. So I think there's much more opportunity in these kinds of environments than we have in Norman. Yeah. Well, I think people have moved to precision as a way of, as in preference over personalized medicine, but I think they're after the same idea of being able to individually tailor therapies. The problem is that the information is generated often at a population level, right? And so we're not, I think, and we can't get the sequence data into practice. So. Huh. Huh. And then we got a question over here. Yeah. Yeah, well, so I like that suggestion to back up of that, because the attempt is to make precision synonymous with personal, but I think you're right. It has a tendency to work at cross purposes to that, especially if you're you just have the same 15 minutes, right? And it's suddenly crowded with a bunch of sequence data as well. You know?
Like the one I forgot to mention, Paul asked Peter Blue is just become president of the board of an organization called Zero Three for infants, children, and families. Part of the dialogue is how to have more practice-based evidence.
Yeah. 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 So I don't know about you, but when I go to see my primary care doctor now, he's put the screen in between me and him, so he can at least look at me while he's filling out the electronic medical record. It used to be he wasn't even looking at me, right? So uh, but yeah, I think that I like the molecules to meaning. I think because that's I think in part what you're talking about is the texture of individual lives and the sense that they're making of what's going on. And yeah, you may have some opportunity to explore that in psychotherapy, but in primary care, you certainly don't. And under this fantasy, you won't. The computer's going to do that work for you. So I think that yeah, there's certainly probably something wrong.
Are we doing well enough in our training in the area of how to use video?
Oh, I think most people in medicine would say no, right? I don't. Right. That's what I meant. That's what I should have said, right? Right. Yeah. I think that's I mean, it's long been a challenge in thinking about how any of this information gets to be implemented in primary care, let alone specialty care. Yeah, I don't know. I don't know in terms of time. Yeah, but farmer. Yeah, yeah, yeah. Yeah. And I will say, just before you go, if you don't mind, the one place where there has been real progress in doing genetic work in tribal communities has been in Anchorage at South Central Foundation at the Alaska Native Medical Center in the context of pharmacology. Yeah, yeah, yeah. Hmm. Yeah. 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 Right. Yeah. That's, and I think that's and my apologies if that wasn't clear. I think that has been the revolution, right? Is just an awareness of how these systems are working and how genes are interacting. It's changed the way we think about biology, I think, in fundamental ways. But yeah, the clinical utility has been harder to realize as a consequence of that complexity. Yeah. Yeah. Right, right. They do change. Yeah.
Analogy to the G factor in intelligence, P factor, they're thinking about extracting a number of G for intelligence for all of psychopathology.
Oh,
Just to polarize it, as you say,
well,
because of that, so it's very thoughtful
so I don't know in terms of time if you want to wrap up or keep going.
usually have the opportunity to go
All right.
for now and then.
All right. All right. I don't want to cut it short. It's getting interesting.
That's what it's
But I may have just done that. Oh, good. Thank you. Yeah. 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 No, because I'm right. Right. No, I mean, what I'm thinking about is a possibility for advancing knowledge about what goes wrong in people's lives that is opened up by these catalogs that we can now look at. But no, I'm not an informaticist. I have no specific proposal there. Again, I don't want to put the one we have in the room on the spot.
bridge that gap, get better context.
Well, yeah, we've got more data than we know what to do with. Yeah. Well, that's, as I said, that's the challenge my colleagues. Yeah. Yeah. Well, they no longer will give you the money just to find a pattern, right? Yeah, they are interested in, at least that's our experience. They're interested much more in, understanding what's going on. Mm-hmm. Yeah, and certainly the NIH is as well. Yes.
We have genes. So one of the few things that are positive in the United States now is genetic information. So you're helping get well, only involved. All right, but nobody's yet. Right now, although our genes suggest we all have pre-existing the health insurance can't discriminate.
Yeah, oh, if you participate, I mean, you not only are paying for the negligible report you get back on your ancestry, but you are giving them the rights to all of that information, right? To do with as they see fit. Yeah, 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 yeah. And they potentially they have the whole sequence available. I don't know if they've actually generated it. Yeah, right. But they still have the samples, right? Yeah. Yeah. Oh, yeah. Sure. Well, welcome to Colorado. On both sides, right? Yeah. Yeah. Right. And those kind of archival records that you would need to be able to capture that process in terms of the biological materials, yeah, I don't think that that's contemplated in all of us. There's a reason the sequence data is appealing, right? Because it doesn't change, right? Yeah. Well, before we come back to that where you've ended up, I want to come back to that suggestion you made, which hadn't occurred to me before you said it, about the gap in terms of practice, in terms of integration with social and behavioral expertise as well. 0 to 3 is involved in an effort to take an intervention called Healthy Steps to scale nationally. And what that effort involves is embedding expertise in child development in pediatric practices. And the idea is that ultimately that would be available to everybody, which rather than relying on the poor pediatricians who are already plagued by a bunch of demands and really want nothing to do with any of us who have an additional suggestion on something else they could do in their time with the patient. It creates a supplement, a complement to that on the side of social and behavioral expertise that would allow for parents to talk about concerns around sleep, around crying, those kinds of things. But with regard to the other question, again, I'm going to defer informaticist.
Well, so that's why I'm pushing on proteomics, because there are elements that change. Like genome doesn't change much. And probably is going to change much dynamic. Although there are specific. But there's plenty of things that you can interrogate at the genomic level. RNA for expression, protein, that do change. And those changes are big. And I think the real challenge is the developmental the changes that happened in utero or 20 years ago, whatever, and have lasting effects. And that's what, I don't want to take issue with you calling all of us lunch.
Yeah, for the biological. Yeah, yeah, right. No, yeah, exactly. And I think that's true for almost all biobanking, right? Yeah. It would be unique. It would be unique. Yeah, I think. Yeah. Yeah.
So I'm curious about the comment you made about cancer cells being
Oh, no, no, no. Just that they have them both, but they're like separate worlds, right? So cancer prevention and control, population sciences, that's all of us looking at epidemiology and preventive intervention, social and behavioral preventive intervention. And then you have the people looking at tumor genetics and molecular processes, and we don't really often talk to each other.
there is a growth of cancer programs introducing a whole set of psycho-oncologists within those programs to help support as well as the patient, which may or may
But
not.
does that get back to the molecular work though? No, certainly survivorship and palliative care, and that's a part of that social and behavioral portfolio of most cancer centers. But what I was talking about is the dialogue between the molecular work and that social and behavioral work.
Susan particular is a person she's a psychosocial oncologist who works with a cancer researcher looking at activation of TNF alpha, for example, within the tumor itself affects proliferation of tumor cells. So there are
Yeah.
a few. So it's
And
starting
yeah,
to use
yeah. And you,
is
no, it's just a persistent tension is all I was saying between those two worlds of science. You know, as I said, from the basic science perspective, I'm not even a scientist, but then it just slips out. That's how they see us, but they don't see us as scientists. But you remind me, we just recently had a talk from a survivorship researcher who is studying physical activity in survivors primarily, the motivation is to improve their quality of life from a clinical perspective. That's their motivation. But the NCI's motivation for funding that work is to study the impact of activity on cancer recurrence, which is an example of taking then something social and behavioral and linking it to a biological process that is recurrence of cancer. So yeah, I think there are examples, certainly. I just meant to illustrate that when we were site visited and preparing for our site visit, it was a 20-year process. We finally got NCI designation. That was just clear that every cancer center struggles to not to host a cancer prevention and control survivorship research program and a basic science research program, but to facilitate dialogue between them. Yeah. Yeah. Huh. Yeah. All right. I think that's good enough. Oh, I'm sorry. Ted had a question. Ted had his hand up. Yeah. Sorry. He's clapping though, so maybe. Yeah. 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 Right. Yeah. 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 Right. Yeah. There's lots we know that we don't act on.
Poverty, violence,
Mhm.
and that's what you're going to take up next.
Sure.
Join you, thank you, Paul, and hopefully council goes to weekend, grand rounds tomorrow.
Yeah. Give me lots of new ideas. Yeah.
So thanks, Paul.
Yeah. Thank you.